This video is meant to describe and explain what you need to do for writing your first lab report. The first thing I need to tell you is that your teaching assistant or instructor has given you, in many cases, a specific rubric or a set of directions of what they wanted. If they haven't already done that, they will be doing it very soon. The most important thing that I can say probably in this video is that you need to be following those instructions as precisely as possible, because those individuals are the ones who are ultimately going to be grading your work. Now, if they haven't given you a rubric or some kind of directions yet, wait patiently, because they will be doing so most likely in this coming week. All right, so that being said, let's talk about what's happening in the lab and what kinds of things you need to be writing in your lab report. So you use the colorimeter and the phenomena that controls particles in a solution and their ability to absorb or transmit light, which is described by Beer's law, to build a standard curve. And that standard curve is a tool that you then use to find the concentration of an unknown starch. And your TA may ask you to include that in your lab report or not, but when you come then to the next lab, the lab you did using amylase, where you use then the colorimeter that you knew already what to do, you use that colorimeter then to observe amylase's function. Now, amylase is not something that I can show you in person. I can't say, here's amylase, like I can say, well, here is a chicken, or this is the skeleton of a mouse, or this is a slide that shows a red blood cell. Those are things that I can show you, but amylase is not something that I can show you easily in this laboratory. But we can see the function of amylase as it breaks down its substrate. So amylase is the enzyme, and it does not change over the course of the reaction. But the potato starch, which is the substrate, does change. It breaks down from its whole self, from the potato starch, into then other molecules, monosaccharides or disaccharides, over time as amylase does what it does. Okay, so amylase, by the way, is found in your body, in your saliva. So it's produced by the glands that you have in your mouth and the back of your head. It's also produced by your pancreas, and in both cases they break down carbohydrates, not just potato starch but other kinds of starches. And amylase has a condition in which it optimally functions, and I would suggest that it's important for you to know what those conditions are. So in other words, enzymes operate optimally under certain temperatures, under certain salinities, under certain pHs, even pressure, at certain pressures. So there are things that are going to allow amylase to break down starch more efficiently than other conditions or other things. Things is a horrible word, so we'll use conditions. It's a better word. And you can look up that information online. Now, your instructor may be requiring you to have a formal reference section, and if they are, then you need to make sure that you very formally reference the things that you find about amylase. If you are not required to have a formal reference section, that's still fine. If you make a statement about amylase, you didn't just know that, you got it from somewhere. So even if all you do is provide the name of, the webmaster or the author of the paper that you read and the date that they posted the last information or that they printed the article, that would be something that is minimal. You can't make statements without providing some evidence of where you got that information from, okay? Especially when you're talking about something like optimal function of amylase. That's not common knowledge. You need to note it. So once you have got that, you have to have some kind of hypothesis. You would have had a hypothesis last week when you did this experiment. So amylase ran at an ambient temperature, which means room temperature, just the pH of the water that you put the amylase in, so close to 7, certain salinity, certain conditions, You measured those to start with, ambient conditions, and then you changed those conditions, and you had a hypothesis about what would happen. Would the absorbance or the transmittance increase or decrease based on the conditions that you changed? Now, you then change the conditions and you have the graphs. So what do you need to do to report? Well, we've got our hypothesis that belongs in the introduction, and anything that you put down in terms of amylase's function also belongs in an introduction. How formal of an introduction you need is going to be determined by your instructor. You need then a method section, and the method section describes what you did in precision. It does not describe what you were told to do by the lab book. It does not describe what you wanted to do or what you thought you should do. It says, what did you do? So if you copy down the directions from the lab book, then you're not going to get all the points, you're going to get a bad grade, or you're going to be asked to rewrite. One of those three things is going to happen. You need to be telling your reader what you did in past tense, and that's how it should be written. Okay? In my case, when I ask my students to turn this in, I do not ask for a list of materials. I expect for those materials to be written in a paragraph form. So if you're describing, the colorimeter was used for the purpose of determining the rate of potato starch breakdown by amylase, that tells you several of the things that were required without having a list of materials. I also think you should use formal language. Pronouns do not belong in lab reports, so the word I or we, not something that you should be using. Avoid pronouns as much as possible because it will help you write a better lab report. So after you have your method section, then you're going to have your results. Now the results, the raw data, is the same for everyone. You should take the raw data, you should go separately and produce your lab reports. If you do, even the graphs, even though the curves may be the same, the details of them will be different. You are not allowed to print one lab report and then share it amongst 2 or 3 or 4 people. It's not how it works. The raw data is shared and that's it. So you need to be managing your raw data. It needs to be presented in tables or graphs, whether they be line graphs or pie graphs, some form of a graph. You can present them in some form that is organized and that is conveying meaning to your reader. So let's say you produce a graph, which in this case you will be doing, probably a scatter plot that has a line and then a trend line. You need to describe that graph. So first of all, you need to label X and Y axes. You need to have the formula for slope that's posted. You should already know how to do this. If you don't know how to do it, I'll tell you in a minute where you can find out. Okay, you need to have a label for your graphs or a title for your graph. These are things that you knew in high school. Let me remind you. Okay, and then after your graph is properly labeled, you need to have a legend that describes that graph and what it is that your reader is looking at. That does not complete your reference section. Your results section. Your results section still then has to have a description. This is what the results are in a paragraph form. If there's some repetition between the legends and between the paragraph that describes the results, that's okay. It's better to have repetition than it is not to have a full description. Okay, now back to this idea of how do you put a trend line, how do you produce the graphs in a way that is appropriate. If you're not already familiar and you haven't been shown how to do that, or you were shown and you've forgotten how to do that, there's a place where you can get help. You can go to the mentors, the biology mentors, and they are going to be in room 106 of BO Hall. That's a student lounge. And in the back of that, there's a small room that's 106C, and that's where the mentors are waiting. They sit there, they volunteer 2 hours a week and sit there and wait. So there's 43 different people who go in there and wait to help students do what they need to do in biology. And so helping you with these graphs is something that they can do. You need that trend line and you need the equation for slope, because comparing the slope is what you can do to compare your ambient condition to your experimental condition. And if that statement doesn't have meaning to you, you need to go see the mentors, or you need to talk to your instructor. All right, that having been said, once you have your results, then you need to move to conclusions. This is really a hard part. This is a part where we get to see you think, because what you're doing now is taking this data that you've organized and you're saying this data supports these conclusions. These things are true of amylase. These, well, not true, these things are supported that amylase does this, and the support that I have for this is the results that I gained. Okay? So as a result, the hypothesis, what I thought was going to happen, the answer to the question that I provided, the question being, how does amylase behave under different conditions? I answered that. Either that answer was supported or it wasn't. Okay? Now, understand that you can go too far with a conclusion. You can conclude too many things. For instance, you can conclude that amylase is the best enzyme to break down starch. Well, you can't say that in these conditions, in this experiment, because you didn't compare another enzyme. But you're not going far enough if you say, well, amylase worked at 72 degrees Celsius and it didn't work at 55 degrees Celsius. Therefore, amylase only works, no, 55 and 72, that's not, I'm thinking Fahrenheit. Let's try it again. Amylase worked at 27 degrees Celsius, but it didn't work at 55 degrees Celsius. And therefore, amylase only works at 27 degrees Celsius. Well, that's not looking at all of the conclusions, because what about 30 degrees Celsius or 37 or 40? You got to make sure that you say everything you can say, but not more than what your results support. So it's a little bit tricky, and chances are you might not get it exactly right the first time, but that's okay, because your TA will have some kind of an idea of what they're going to do to help you with that. Either they'll ask you to rewrite to regain some points, or they may grade very easily the first time and then get harder as the lab reports go on, because you will have other lab reports to produce. But they'll help you understand, okay? You do want to do a good job and get the points from this for two reasons. One, you want the grade, but two, you really need to understand how to do this. Lab reports are something you're going to have to produce in the future, so learn how to do this and do a good job. Again, I remind you, the number of references, or if you need a reference section, how extensive the introduction needs to be, and what kinds of things your instructor is looking for. Those are things that they will give you in either a rubric or a set of directions. Make sure that you follow those very carefully, because they know what they want. Okay? And I hope you have a wonderful evening and you enjoy next week's lab, which there is a video put up which is on osmosis and diffusion.